So Bryan's going to present today the result of his master's studies, his completion seminar. Bryan came to us a few years ago from Peter Mac, having studied the effects of drugs on humans, and was very interested in understanding how drugs work together in combination, how combination therapies might be made more efficacious, possibly by using quantitation and modern techniques. Sue and I were interested in that idea anyway, so it was fabulous to have someone come to us with the same idea. He's embraced it, he's tackled it, he's done a brilliant job of the experiment, as you'll see, and presenting some very nice and very challenging and very inspiring, I hope, data that encourages us that we're on the right track, as we'll see. I should tell you that Bryan has taught Sue and I something, which is that we have to be very careful of the exact words we say, because he takes us very literally. So for example, one example is, he showed me one of his beautiful plots one day, and I said, that looks like a cockatoo, that's a cockatoo plot. And a week later we were reviewing data again, and Bryan said, I couldn't find any description of how to plot the cockatoo plot. Where did he get that from? So, maybe look at the cockatoo plot today. I think it's in there. Please welcome Bryan for his completion seminar. Bryan, over to you, if you're ready.
Thank you, Doctor. Hi everyone, thank you for coming to my talk. So today I'll be presenting the work that I've done for the past 2 years. So to start off, during infection, when T cells receive recognize the antigens in the context of MHC peptide, they become activated and they will undergo clonal expansion. So they expand into a large population of effector cells and memory cells in order to fight the infection. So once the infection is cleared, they undergo a contraction phase, as you can see. So they undergo a contraction phase, and in the end, what you get is a pool of memory T cells. So the magnitude and the duration of the response is determined by the number of cells that are responding, the rate of division, the number of division rounds the cells undergoes, as well as the rate of the cells die. And all in all, this whole immune response is amazing because it not only helps us clear infections, but also ensures a long-term protection against reoccurring of the infection. However, this response can backfire at us in the context of autoimmunity and organ transplantation. And therefore, we need to employ the use of immunosuppressive drugs to control the responses. Now, the challenges with immunosuppressive drugs is that even though the mechanism or the molecular targets of most of these immunosuppressive drugs are known, we actually don't know how they're actually suppressing the immune response. In terms of how, we don't know how they're affecting the rate of division, we don't know how they're affecting a number of divisions, the rate of death, and number of cells in order to get this suppressed response. So the idea for my project is that we can utilize the quantitative methods that are developed by the lab to understand these whole underlying processes of survival and proliferation kinetics. So the hypothesis of my project is that we can utilize the quantitative methods to quantify the effects, the individual effects of immunosuppressive drugs, so isolate the effects on the survival and proliferation. So the second hypothesis we have is that once we're able to quantify these effects, we can utilize these profiles and be able to predict the effects of 2 or more immunosuppressive drugs in combination. So in order to achieve these, to test these hypotheses, for my current project, we have 2 aims. So the first aim is basically we quantify the effects of the immunosuppressive drugs individually, and then next utilizing this, the profiles that are generated, we also then quantify the effects of the immunosuppressive drugs in combination and develop rules to be able to test their effects in combination. So our lab have utilized and standardized the OT1 and the OT1 BIM knockout system to study the survival and proliferation kinetics of CD8 positive T cells. So for our methodology, we label the cells with a proliferation dye known as cell trace violet, and then we stimulate them with the N4 peptide, which is chicken albumin, that's recognized on the OT1 receptor of the CD8 positive T cells. And then we also introduce a co-stimulating molecule, which is anti-CD28. And also, not because activated CD8s can endogenously secrete IL-2. So what we do is we actually block the effect. And then what we do is then we culture the cells and then we harvest these cells at various time points. So these are the cells at 27 hours. As you can see here, the cells have not divided. And then over time, across the different time points at 40 hours, 64.5, and 73 hours, you can see that the cells start progressing through divisions. So then what we do with this raw data, or this time series data, is then we measure the number of cells in each division, and then we plot it over here. So we plot it again for each time point, the cell numbers in each division. And then what we do next is then we analyze the results using the quantitative model that we have, which is the Cohort Method, which will be what I'll be describing today. So what is the Cohort Method? So the Cohort Method aims to basically correct for the effect of cell expansion. As you know, when one cell divides, it becomes 2, 2 becomes 4, and 4 becomes 8, and 8 becomes 16. So we try to correct this by dividing the cells in each division by 2 to the power division number, and we get the cohort number. And cohort number refers to the number of founded cells that contribute to the population that we get. So with this cohort number, we can extract various informations. So for example, if we were to calculate the total cohort numbers for every time point, and we were to plot it against time, we can get the rate of death over time. And if we take a time point before the cells divide and normalize that to 50% of that and extrapolate it back to the graph, we're able to calculate the mean time it takes for the cells to die. And then if we were to calculate the average number of divisions for every given time point and plot against time, we can get a plot of mean division number against time, where if we were to extrapolate the graph from the mean division number of 0.5, we are able to calculate the time it takes for the cells to enter division. And basically, the division rate, as you can see here, gives us the time it takes for cells to progress through division. And then when the mean division number plateaus, this refers to as, we calculate this as division destiny, which is the maximum number of times the population divides before they stop and reach quiescence. So these are the examples of immunosuppressive drugs that I'll be presenting today. So the first drug we'll be talking about is rapamycin, which is used for the treatment of organ transplantation. So it targets the FK binding protein 12, which downstream of that inhibits the mTOR1 signaling pathway. Now, one of the effects of rapamycin is that it reduces proliferation, and that it can reduce the activation of T cells. Another drug that I'll be showing you now is Mycophenolic acid, which is used to treat organ transplant, used for the treatment of organ transplantation rejection. And basically, it targets the enzyme that's involved, that's involved in the synthesis of guanine nucleotides, which is the inosine monophosphate dehydrogenase, an S-phase inhibitor. So the effects of mycophenolic acid is that it causes cell cycle arrest. So the next drug I'll be talking about is the class known as the corticosteroids, where examples are prednisolone and dexamethasone. So these drugs are used for the treatment of lupus, erythematosus, rheumatoid arthritis, as well as psoriasis. So these drugs target the glucocorticoid receptor, and what they found was that it can trigger death by apoptosis, which is what they found in the lymphoid cell lines. So another drug we're talking about is the clinical drug, which is the ABT-737, which is the in vitro form, and the ABT-263, which is the oral form. And these drugs have been used in clinical trials for the treatment of lymphoma. So these drugs basically targets the pro-survival molecules, such as the BCL2, the BCL-XL, and the BCL-W. And basically, what they find is that you can get death by apoptosis. So the last drug that I'll be talking about, we'll be presenting, is the Cyclosporine A, which is used in organ transplantation. And basically, they target the calcineurin, they inhibit the calcineurin, and downstream of that leads to the inhibition of the NFAT pathway. So it's found that they can cause reduced proliferation, and they can decrease the survival of activated T cells. So here we have mycophenolic acid, and on the right we have rapamycin. So as you can see, both of these drugs can decrease the total cell numbers in a dose-dependent manner. But the question is, how are they doing this? Are they affecting survival, or are they affecting proliferation, or are they having effects on both? So when we first explore the effects on proliferation, you can see that in dose-dependent manner, mycophenolic acid increases the time it takes for cells to enter division. It also decreases the rate of division, and it also decreases the number of times cells divide in a dose-dependent manner. Rapamycin, also as you can see, increases the time it takes for cells to enter division, also increases the time it takes for cells to progress through division, and also decreases the number of times cells, the number of times the population divides. And if you were to look at the effects on survival, you can see that mycophenolic acid has no effect on survival. And if you were to look at rapamycin, you can see they also have no effect on survival. So this is a really nice illustration on how these two drugs only affect proliferation and has no effect on survival, and yet you can get such a huge effect on the total cell numbers just by affecting proliferation. And if you were to enumerate these values into the time it takes for cells to divide and the subsequent division rate, you can see that mycophenolic acid and rapamycin increases the time it takes for cells to enter division. And also for subsequent division, you can see that with increasing doses of mycophenolic acid and rapamycin, they increase the time it takes for cells to progress to division. So if you were to look at the effects on the number of times cells divide, so you can see that in a dose-dependent manner, mycophenolic acid decreases the division destiny, and you can also see the same for rapamycin. So note that the red line is basically no drug. So this is the time it takes for cells to die. You can see that mycophenolic acid has no effect, and you can see that with rapamycin also it does not affect the time it takes for cells to die. So the next two drugs that I'll be showing you both are dexamethasone and ABT-737. So as you can see, both of them decreases the total cell numbers in a dose-dependent manner. So if you were to explore the effects on proliferation, you can see that dexamethasone there's not much of the effect on proliferation. And if you were to examine the effects of ABT-737, you can see that there's no effect also on proliferation. So if you were to examine the effects of dexamethasone on survival, you can see in a dose-dependent manner, there's a decrease in survival, starting about 50 hours. And on ABT-737, you can see that from about 70 hours, it also decreases the survival in a dose-dependent manner. So if you were to, so this is also another nice illustration, like the previous two drugs I showed you, they only affected proliferation. You have two drugs here that only affects, the only effect they have on the cell number is on survival. So if you were to enumerate that, you can see this is the effect of dexamethasone on the time of the first division, and the effects of ABT-737 on the time of the first division. You can see that both of these drugs have no effect on these parameters. And if you were to examine their effects on the subsequent division rate, you can see that there's also no effect. If you were to, and then when we examine the effects on the number of times cells divide, you can see that dexamethasone doesn't affect the number of division destiny, nor does ABT-737. So finally, when we look at the times to die, you can see that in a dose-dependent manner, dexamethasone decreases the time it takes for cells to die, and you can see the same as well with ABT-737. So very nice illustration of a group of two drugs that only affects proliferation and two drugs that affect survival. So next, we're wondering what does cyclosporine do? So here's cyclosporine. So here's the total cell numbers over time, and you can see in a dose-dependent manner, cyclosporine rate decreases the total cell numbers over time. So if you were to explore the effects of cyclosporine on proliferation, you can see that really nicely that cyclosporine increases the time it takes for cells to enter division. it also decreases the subsequent division rate and also decreases the number of times cells divide. And as you can see, these two concentrations, they don't even enter division. If you look at survival, you can see that dose dependently, they are also affecting survival. And in fact, these two doses, 200, 400 nanograms, they have very huge effect on the survival. So if you were to enumerate these values and have a look, you can see that Cyclosporine A increases the time it takes for cells to enter division. It also increases the subsequent division rate. And note that I was not able to calculate the effects on subsequent division rate following in the later concentrations. And also decreases number of times cells divide. And finally, it also decreases the mean time it takes for cells to die in a dose-dependent manner. So overall, what I've shown is that, I've shown that I've been able to identify the mechanism of how immunosuppressive drugs can inhibit the expansion of the cells. So I've shown you some drugs that only affects survival, and I've shown you some drugs that affect proliferation, and I've shown you cyclosporine, which affects both. So this is a really nice, this also is a really nice illustration on how we can categorize these drugs into different groups. So you have rapamycin and mycophenolic on proliferation, ABT-737 dexamethasone on survival, and cyclosporine on proliferation and survival. And these two are really, as I said, a really nice illustration because we get drugs that only affect one side of the parameter, like drugs that only affect survival and drugs that affect proliferation, and they don't seem to be. And these two fates don't seem to be interfering with one another, which nicely suggests an independence of the two fates of survival and proliferation. So next we explore the effects of these drugs in combination. So if we were to look at drugs that affect proliferation, rapamycin and mycophenolic acid, and combine together. So here we have mycophenolic acid alone. So in those dependent decreases cell numbers. And I've shown you that it affects primarily on proliferation and no effect on survival. So here we have rapamycin alone, and I've shown you that it only affects proliferation and does not affect survival. So if you were to combine them together, so this is mycophenolic acid at 50 nanograms per ml, titrated with increasing concentration of rapamycin. You can also see that with increasing rapamycin concentration, you get a decrease in the total cell numbers over time. And you see that as well for mycophenolic acid at 100 nanograms per ml, where you get decreases in total cell numbers with increasing rapamycin concentration. So if you were to examine their effects on proliferation, you can see that dose dependently, rapamycin decreases the number of times cells divide, as well as also increases the time it takes for cells to enter division. And if you were to increase the mycophenolic acid concentration, you still see the same effect of by rapamycin. However, if you were to look at effects in survival, you can see that in combination, rapamycin doesn't change that, or doesn't further enhance, or nor does it decrease the effects. Mycophenolic doesn't decrease the effects on survival, and you do not see also any differences at 100 nanograms per ml of mycophenolic acid. So, really, what I've shown you is that in this combination, survival is not affected, and proliferations are only affected. But the question is, how are these? Are their effects in combination additive, or are they just or are they synergistic? So we explored various rules on how we can calculate the effects together. So the first rule we tested is the additive rule. So if mycophenolic acid at 50 nanograms per ml decreases the number of times cells divide by 0.5, as you can see here, and if you have rapamycin that decreases the number of times cells divide by about 1, we expect that the combined effect should decrease the number of times cells divide by about 1.5 times. And then, so this is the result when we measure. So this clearly shows how independently these two drugs add together to inhibit the number of times cells divide. So next, I'd like to show you combinations of drugs that affect different parameters. So drugs that affect things, proliferation, and drugs that only affect survival. So here we have rapamycin alone, as I've shown you before. So they only affect proliferation and have no effects on survival. Here we have dexamethasone, and they have no effects on the proliferation, and they only affect survival. So if you were to combine them, you can see that with increasing doses of dexamethasone, you get decrease in the total cell numbers. And if you were to explore that effects on the proliferation, you can see that with increasing, that dexamethasone does not enhance the effect of rapamycin on proliferation. And this effect that you see is only by rapamycin alone at 158 picograms per mil. If you were to look at the effects of rapamycin at 150, you can see that dexamethasone also doesn't enhance the effects on proliferation. So if you were to examine the effects on survival, you can see that for both of these, you can see that there is a dose-dependent decrease in survival by dexamethasone. So if you were to enumerate these values, we can see that, so this is the effect of rapamycin alone. This is the effect of dexamethasone alone. So you can see that rapamycin increases the time it takes for cells to enter division, and dexamethasone does not affect the time it takes for cells to enter division. And if you were to combine them together, so note that the red line is now no drug, and then the x-axis is just dexamethasone with increasing concentration. And then if you were to, and then the color represents the concentration of rapamycin used. So if you were to increase the concentration of rapamycin, you can see that the time to first division still is unaffected by the increasing doses of dexamethasone. If you were to use, now at rapamycin 500, you can see that the effects on time to first division also is not enhanced or not affected by dexamethasone. So if you were to examine the effects on the drugs on the number of times cells divide, you can see that rapamycin decreases number of times cells divide, and dexamethasone has no effect. And in combination, you can see that if increasing doses of rapamycin, dexamethasone doesn't change that. If you were to explore the effects on the survival, so the mean time to die, or the death time, you can see that rapamycin does not have an effect on the death time, and I've shown you before dexamethasone decreases the death time in a dose-dependent manner. In combination, you can see that even with increasing doses of rapamycin, the death time does not change. So it's really, really nicely showing that the effects of these two drugs on the survival and proliferation are independent. So next, we want to talk about how we wanted to explore the effects on the of these two drugs on the population level. So before I go on to that, I'd like to take us back and try to run through an understanding on what happens in the culture. So in culture, you know that a lot of things happen simultaneously. We know that there are cells that are dividing, there are cells that are reaching quiescence, and there are cells that are dying. And all these processes are unsynchronized, which makes it really difficult to predict how drugs can have an impact on these values. However, when you have two drugs that act on different outcomes, so you have a drug that only affects survival and a drug that only affects proliferation, predicting it, we can treat the actions on the outcomes as independent, and then we can actually be able to predict them or become more predictable. So it means that these simultaneous effects that are happening with these two drugs is equals to the effects that we apply sequentially. So in order to illustrate my point, I have diagrams to show you. So here we have drug A, and here we have drug B. So drug A decreases the total cell numbers by 0.44, and drug B in comparison to control is 0.34. So if you were to add these two drugs together, what we expect is that the effect of drug B should be added to drug A, and basically you get a 0.34 decrease on the cell numbers of drug A. And this is what we expect. So this will be what we expect of the combination of drug A and drug B. So if you were to do the fold change difference, you'll get a 0.124 difference between the control and the combine of drug A and drug B, which is equivalent to multiplying these two fold changes. So to illustrate what I've done, so this is rapamycin at 500 picograms per mil. So the black line is control, and the purple line is rapamycin at 500 picograms per mil. So if we were to do a fold change over time, so basically we normalize everything to the controls at different time points. So controls will be 1 at every time point. And if you were to normalize the rapamycin at every time point to the controls, this is the fold change that we'll get. And then if you were to normalize it to for dexamethasone as well, this is the fold change we get in dexamethasone. And then what we predict. And then when we multiply these two effects, this is what we'll predict. So if we were to apply this predicted fold change onto the control, so this is what we expect the total cell numbers to be. And in fact, this is what we measure. So really nicely showing that, that these two effects of the rapamycin and dexamethasone on the population level is additive. Now, another combination I'd like to show you guys is a combination of mycophenolic acid. As you can see, only affects proliferation, no effect on survival, and dexamethasone alone, which has no effect on proliferation and only acts on survival. So if you were to combine these two drugs together, so here we have mycophenolic acid at 100 nanograms per ml, titrated against dexamethasone, and here we have 200 nanograms per ml against different concentration of dexamethasone. So if you were to look at the effects on proliferation, you can see that dexamethasone does not change the effects it has on the times to divide as well as the number of times cells divide. You can see that at mycophenolic acid at 200 nanograms per ml, there's also no difference or no effect by dexamethasone. So if we were to explore the effects of dexamethasone at mycophenolic acid at 100 nanograms per ml, you can still see that decrease in survival by dexamethasone, and you also see that at mycophenolic at 200 nanograms per ml. So if you were to enumerate these parameters, you can see that, so this is times the first division. So mycophenolic acid increases the time it takes for cells to enter division, and dexamethasone does not have an effect on the times the first division. You can see that in combination, in a dose-dependent, with increasing mycophenolic acid concentration, dexamethasone does not have an effect on the times to divide. If we were to look at the effects on division destiny, so this is mycophenolic acid and this is dexamethasone, you can see that in combination with increasing mycophenolic acid concentration, dexamethasone has no effect. If you were to look at the times to die, so this is mycophenolic acid, which has no effect, and dexamethasone, you get a dose-dependent increase. You can see that in combination, mycophenolic acid, does not enhance the effects of, does not change the effect of dexamethasone on death. So really nicely, another nice illustration that, this of two drugs affecting, causing different outcomes to be independent on the parameters. So if you were to explore the effects on the population level, so this is the fold change of dexamethasone and the fold change of mycophenolic acid. So if you were to multiply the fold changes and create a prediction, this is what we predict the fold change will be. And if you were to apply that to a prediction on the cell numbers, this is what we get when we measure. So they are really nice illustration that the effects of these two drugs on the cell numbers are additive. So another combination, rapamycin and ABT-737. So here's the effect of rapamycin. So only affecting proliferation, no effect on survival, and ABT-737, which is a BH3 mimetic that only affects the pro-survival molecules. So as I showed you before, ABT-737 has no effect on proliferation, and its only effect is on survival. So if we were to combine these two drugs together, so this is rapamycin at 158 picograms per ml with increasing ABT-737 concentration. And as you can see, there's a dose-dependent decrease in the total cell numbers by ABT-737. And at rapamycin 500 picograms per ml, you see that same effect as well. So next you want to know is how are they affecting proliferation? So, as you can see, ABT-737 has no effect on the proliferation, so the effect's only by rapamycin. And at rapamycin 500 picograms per ml, ABT-737 has no effect, and the effect you see is only by rapamycin. So what about survival? So if you were to explore the effects on survival for both concentrations, you can see that the survival is only affected from about 70 hours onwards. You can see the same here, you can see the same here, and yeah. So which nicely shows that these effects, also like the other two, could also be independent. So now if you were to enumerate these effects on the time to first division, you can see that the effects on time to division, on times to first division are independent. So increasing rapamycin concentration, ABT-737 does not enhance the effects of rapamycin on the times to first division. If we were to explore the effects on division destiny, you can see that rapamycin decreases division destiny, and ABT-737 has no effect on division destiny. And in combination, you can see division destiny is not affected in combination. So if you look at the death times, so rapamycin, ABT-737, which decreases, and you can see that there's no enhanced effects on the death time. So like the other two, this is another nice illustration of how these two drugs are acting independently on their own respective parameters in combination. So if we're to explore their effects on the population level, so the red line is ABT-737 at 1 micromolar, and the gray line is the rapamycin at 158 picograms per mil. So if we were to predict the fold change and apply that on the cell numbers, and then we plot it with a measure, you can see that they're almost identical. So really nicely showing that even on the population level, the effects are additive. Finally, the last combination I'd like to show you is mycophenolic acid and ABT-737. So here's the effect of mycophenolic acid alone. So they only have effects on proliferation and no effect on survival. And ABT-737, which has no effect on proliferation and only affects survival. So if you were to combine them, you can also see that you can still see the effects of ABT-737 on the total cell numbers. And if you were to explore the effects of proliferation, you can see that that effect remains unchanged. So the effects on proliferation is only by mycophenolic acid at 100 nanograms per mil. And if we look at the effect on survival, you can see that survival seems to be only affected by ABT-737. So if you were to enumerate these effects, you can see that, so this is the effect of mycophenolic acid on the times to first division, the effect of ABT-737 on the times to first division, and the effects in combination. You can see that the effects are independent. If you were to look at the effects of mycophenolic acid and ABT-737 on the division destiny, you can see that in combination, there doesn't seem to be any changes to division destiny. So if you were to explore the effects on the death times, so it's affected by ABT-737, you can see in combination it's still independent. So really, basically I've shown you all 4 combinations of drugs that affect different parameters. In combination, they still act independently when combined. So, like the others, I was wondering, would we actually still see the additive effect on the population level? So let's explore this together. So, this is the fold change for ABT-737 at 1 micromolar, and this is the effects of mycophenolic acid at 100 nanograms per mil. So if we were to multiply these fold changes together and apply it onto the control, and to create a prediction of what the total cell numbers might be, this is what we got. So another nice illustration of how these two drugs are also adding on the population level. So, to summarize everything, so individually I've been able to identify the mechanism of how drugs can affect the cell expansion in terms of the survival and proliferation kinetics. I've shown you how we can categorize these drugs based on the isolated effects. So as I showed you in the grouping earlier, like 2 drugs in survival, ABT-737 on dexamethasone survival, Cyclosporine on survival and proliferation. And in combination, I've shown you guys 2 drugs that affected proliferation. So with only 1 example so far, I've shown you that we were able to demonstrate that the inhibitory effects of those 2 drugs on proliferation were additive just by using a simple rule of addition. And hopefully this will give us a nice principle that we can apply other simple rules for other combinations. Finally, drugs affect different parameters. I've shown you really nicely that on the survival and proliferation kinetics, they are acting independently. And that behavior is also seen on the population level, where the effects are just added on sequentially on top of each other. So really, the take-home message from my project, really nicely, is that this whole project is a really nice, powerful principle that we can utilize modeling and quantitation to be able to potentially predict the effects of drugs in combination. So before I end my talk, I'd like to thank my supervisors, Phil and Sue, for the support, encouragement, and also their guidance throughout this whole master's. It's been really, thank you very much. And that's all I can say. I'd like to thank Julia Machingo for being such a great mentor, for providing me feedback and guidance, and teaching me how to be able to think critically and analyze data, as well as I learned some new cool stats tricks from you too. Thank you. I'd like to thank Kim Fan and Andre and Manny and Charlotte, Jay, Vanessa, Izzy, and Jessica. So basically everyone in the Hodgkin lab. And apologize if I haven't missed anyone else here. I'd like to thank the animal facility, Lauren, Merle, and Daniel, for without them I wouldn't have any mice for my experiments. Yeah, and I'd like to thank Dimi for the constant service as well as support whenever I had trouble with the facts, as well as teaching me how to be able to operate and use the for TESA really efficiently. And I'd like to thank the Immunology and Molecular Immunology Division for their support. And I'd like to thank Melbourne University for my funding body, as well as Phil and Sue. That's my funding bodies for my project. And yeah, thank you very much.
Thank you very much, Brian. We'll take questions. Do we know how to turn the lights on? Is it wall on, maybe?
Turn it off?
On.
Oh.
So that you can see exactly.
Oh. Okay. Hi.
Hi, that was really cool. It's the first time I've seen that. It's really nice. Well done, Brian. So that data was all generated using T cells.
Yes.
So the obvious question is, do you get the same sort of results with those drugs when you use different cell types? And then, so for example, tumour cells. And so following on from that, if you were to do the same could you use the same approach, say using tumour cells and then testing targeted combinations? Like for example, a lot of chemotherapies rely on proliferating cells, so and Cyclosporine A sort of overcomes resistance to chemo and stuff. So could you use that sort of approach to screen drug combinations?
At the moment, we're still actually studying. So, when I mentioned that, T cells have a division destiny, so that takes how many times they divide. So cancer cells are unregulated, so they don't really have a proper division destiny. They just keep dividing and dividing and dividing. So currently, the Kim Pham, the postdoc lab, is actually exploring this whole competition between the proliferation and the survival, and looking at it really closely on the microscopy level. And she's actually examining the effects on chemotherapy. So I would say, potentially, we could be able to reach the level where we can also calculate the effects of the drugs in combination on cancer cells.
Great talk, Brian. So I guess it seems like it makes more sense to me that the effects would occur independently, where you see one drug affects proliferation and one drug affects survival.
Yeah.
But was it surprising to you that both drugs that affected proliferation acted independently and that their effects were additive? And what do you think that says about what's driving proliferation and those sort of mechanisms?
Sorry, well, can you rephrase the question?
I don't know what
it seems like...
So it seems to me where you've got a drug that affects survival and a drug that affects proliferation. It perhaps makes more sense that when you add them together, that they would be working independently, because it's already possibly obvious that they're working in different pathways, right? But when you have two drugs that affect proliferation and you add them together, I mean, I guess I might have expected that together they would have no greater effect than each one individually because they might have been working on the same pathway, for instance. But you saw an additive effect when you added them both together, which I think is kind of surprising, right? And I guess what I wanted you to comment on what perhaps that says about the pathways driving proliferation. Does it? I don't know. I don't know what the answer is. I just thought that I think it's telling you something about that, right?
Are you telling me asking? I'm sorry.
I'm telling you to answer my question.
First of all, are you surprised?
Yeah. Yes, I was really surprised. Yeah. Because we were surprised. Yeah, we were actually. We weren't expecting them to be able to add together. We weren't expecting the two effects to add together on proliferation. Just like you thought, we thought that it would saturate at a level, or maybe one drug's effect will overpower the other, and you won't see the effect of the other drug. But
so the second question is, with that knowledge, does it give you an insight into the pathway or how it might be working, given that drugs are adding like that? And you can probably just say, maybe.
I'll say that's a very good question. It is. But the thing is, I've only done one combination of testing two drugs affecting proliferation. So I would say I don't have an answer at the moment. But once we're able to test the combination of other drugs that also affect proliferation, then I can perhaps we'll perhaps have an answer or have an idea on what could be happening. Thank you.
I guess maybe along those lines, have you played around with any drugs that you know act in the same pathway? For instance, if there's an ABT that blocks one apoptosis versus several apoptoses, can you pick up the subtlety in the same pathway?
Sorry. Well, can you repeat the question?
So if you pick two, have you played around with any drugs that you know work in the same pathway? And so can you show that there's no additive effect?
That's a good question. I've not actually tested two different types of ABTs in one combination. But I expect that the effects would saturate.
Do it next week.
Sure.
Did you test ABT and Cyclosporine together?
No, not yet.
Because this would be a very interesting combination because it will be a huge synergism.
Hopefully, yeah. So the reason why I haven't tested cyclosporine with the other drugs yet is because we are currently just at the stage we wanted to explore the effects. Because cyclosporine affects both survival and proliferation. So we thought it would be better to explore drugs that only affects one of the parameters. So we have an idea on how they work together in combination and develop rules to be able to test their effects. For example, two drugs affecting proliferation, what kind of rule can we use? Two drugs that affect survival, what kind of rule can we use? And then hopefully eventually we can apply those two rules when we combine cyclosporine with one of the drugs that affect either survival, proliferation, or both.
Because actually, if you have T-cell receptor stimulation, the anti-apoptotic factor A1 goes up, and this mediates resistance to killing by ABT-737 because it's not bound by, it only binds with it's a long break.
Yes.
So if you combine it with cyclosporine, just low dose, you can block the continuity and FAT pathway. And then the resistance of T-cells will be overcome. And you have very strong resistance. So it will be very interesting to test that.
Yeah, exactly. Yeah, you're right.
So you can do that experiment week after next.
I'll just add that now as a clinical issue, we're using drugs in dermatology, except for rapamycin.
Yes.
And the combination of low-dose mycophenolate and cyclosporine seems to be better than just either one of those alone.
Yes.
In low dose though, because we're very worried about toxicity. Yes. So that might fall on there.
Yeah.
My other point was methotrexate is used in psoriasis a lot, and in eczema. These both T-cell diseases that we use these drugs. And Did you test methotrexate at all? Where would you think that would be?
I've actually tested methotrexate. I just haven't presented it here today. Methotrexate effect was similar in the sense to cyclosporine, where it affected both survival and proliferation. And methotrexate is very interesting because the dose window is really, really narrow. So, and they're extremely narrow that immediately, when you reach the cutoff, no matter how much you add, it still saturates and it still causes the same effect on that. So, sorry, do you have a question in regards to that?
And additive? Have you added it with another drug?
No, I've not added methotrexate with another drug because I'm still working on the best on the dose responses of methotrexate. So at the moment, I was only able to have one intermediate dose, and the rest were all saturated. So I'm trying to fine-tune that and be able to identify.
Is that true in the clinic that has a very narrow dose range?
Yes, we usually, in dermatology, we stick to one dose roughly, but rheumatologists go way up before they're They go 25 milligrams a week. We stay at about 10 to 15 for skin effects. It's not high, and not as high as cytotoxic effects in cancer treatment, of course.
Any other questions for Bryan? Jane?
I just had a little question.
Yes.
So you've shown that you did 2 proliferation drugs or proliferation and survival drugs. You haven't shown 2 survival drugs together.
Yes.
So it might tie up your story very nicely.
Yes.
You could use the dexamethasone and ABT because we've shown that it is additive.
That's a very good question.
You've now got 3 weeks' work. 4 weeks.
That's a really good question. My plan was actually to finish up with combine 2 drugs that affect survival, but we actually haven't got to that point yet. So we're still currently working on that. Thank you.
That's it. I think we should congratulate Bryan on a spectacular body of work and wish him well for his final experiments and write-up that's upcoming.